Ladies and gentlemen, thank you for tuning in to the 10th episode of Rowen Reports with Dr. Robert J. Rowen. Today, we welcome to the show the very fascinating Dr. Todd Ovokaitys, an expert on stem cell therapy research. Dr. Todd is certainly one of the most interesting individuals we've spoken with to date, and I'm very excited to bring this conversation to you, our audience. Let us know what you think in the comments of this video, and if you'd like to support this podcast directly, Dr. Rowen is now on Patreon. You can find him there and visit our generous sponsors by following the links in the description below. Now, I'd like to start with some words by Kilroy J. Oldster. Human life is inherently dualistic. It consists of, or is explicable as, two fundamental entities, including rivalries between subject and object, mind and matter, and conflict between the benevolent and malevolent forces. Opposition in the universe creates a dynamic living universe composed of good and evil, body, and soul. Human thoughts and feelings are the communal products of the conscious and unconscious mind's interpretation of a constant flow of coded and symbolic dialogue. Before we jump in, we would like to give a special thank you to our sponsor, PROMOLIFE, bringing you closer to the benefits of ozone therapy. Proudly rooted in the USA, PROMOLIFE is at the forefront of assembling ozone therapy generators designed for both personal and professional use, right here on American soil. Meticulously handcrafted and precision calibrated, each ozone generator speaks quality and reliability. PROMOLIFE has simplified the ozone therapy process by offering a complete line of high-quality accessories that allow you to use ozone in ways you never thought possible. Don't miss out and explore the entire collection of cutting-edge offerings at www.promolife.com/ozone. Make sure you use the coupon code in the description of this podcast for an exclusive discount towards your journey to enhanced well-being. I am also excited that our friends at shopc60.com are helping to sponsor this podcast. If you haven't heard of C60 before, also known as Carbon 60, it is a powerful antioxidant that helps to optimize mitochondrial function, fight inflammation, and neutralize toxic free radicals. I am a big fan of using C60 in conjunction with a keto and intermittent fasting lifestyle to support the immune system, help the body detox, and increase energy and mental clarity. If you are over the age of 40 and you'd like to kick fatigue and brain fog to the curb this year, visit shopc60.com and use the coupon code in the description for 15% off your first order and take control over your health today. This episode is also brought to you by Simply O3. Did you know ozone therapy has been a transformative and medical approach for over 100 years? Used for enhancing immune function, boosting energy, and improving blood flow, it's a powerhouse for maintaining health. Today, over 2,500 medical studies back its benefits, and it's especially effective before workouts, reducing fatigue. Curious to try? Visit simplyo3.com and explore the world of ozone therapy. Use the code in the description of this podcast for 10% off everything they offer, like the Simply O3 3.0 Complete Kit. Begin your journey to better health today. All right, ladies and gentlemen, welcome back to the 10th episode of Rowen Reports with Dr. Robert J. Rowen. Dr. Rowen, I'll let you go ahead and take it away, introduce Dr. Todd here, and get the episode started.
Thank you. I have a very interesting guest for you today, Dr. Todd Ovokaitys. I met him at a meeting of laser therapy more than a month ago in Southern California, and he made a presentation on stem cells that was stunning, beyond stunning, and it shocked both me and Terry to the core. We saw videos of neurologically crippled patients getting better almost instantly. It's impossible to believe, but we had videos of it. And this was he also had a video of a Mexican law enforcement official who was injured in the spine by gang violence, and he was paraplegic, and he was up and walking in weeks. This is something I've never seen before, and I asked him to come on the podcast because his stem cells and what he does is totally different from what the average stem cell physician does, and I want him to explain the difference between the two methods, because if he's got what I think he's got, the man you're seeing should win a Nobel Prize. Dr. Todd, please go ahead and tell us about what you're doing.
Thank you very much, Dr. Rowen. A great pleasure being on with you. I love and respect your work as well. We are in the realm of what we would call laser biophysics, and the work we're doing with stem cells goes across the realm of stem cells and working with a particular type that we believe is generally the most versatile for a wide variety of purposes. So there are other stem cell therapies and different types of cells that are ideal for certain indications, and I'll talk a bit about that. The type of cell that we work with has a really long name. It's called a very small embryonic-like stem cell with the alphabet soup of VSEL. And because we use it so much, I just call them V cells or victory cells or very cool cells. And what's unique about them is that they're formed early in gestation. They're pluripotent, which means they have the ability to make any of the three primary germ layers, endoderm, mesoderm, and ectoderm. And those layers form all the different types of cells of the body. So essentially, it is versatile enough that it can make any type of cell that the body might need. And they're so powerful that they can even make new eggs and new sperm. And we're involved in an IRB approved study. We're actually looking at using them to enhance fertility. It's a very fun project with some very excited potential moms. The hierarchy of stem cells is something I really like to clarify because using the word stem cell is like using the word color. So you might say, well, I'm going to get a stem cell treatment. It's like saying I'm getting a color treatment. Well, there's lots of different colors and there's lots of different types of stem cells. And you know from your work with so-called photobiomodulation that the color or the frequency makes a difference for what it stimulates and what it does. Similarly, the hierarchy of stem cells begins with the early division of cells after fertilization. And up to about 30 cells, it's called the blastocyst. And each of these cells is incredibly powerful. These are true embryonic stem cells, and they're called totipotent. So if you took each of those 30 cells and put it in a receptive womb, you could have 30 whole new people. It can make a whole new person. And this is the one type of stem cell that can have a significant complication, which is called teratoma formation. And teratomas are benign tumors that are super weird because it's a disorganized tangle of all different types of tissues. And if it's big, it could literally be a cantaloupe-sized giant hairball in a space that will fit like the abdomen with skin and hair and teeth and nails and muscle and bone and blood vessels. And that's pretty weird. So although they're typically benign, rarely not, but typically benign, it's not something that we would want a person to develop as a consequence of treatment. Just past totipotent is pluripotent. And that's where the V cells fit. And pluripotent means that the cells are past the risk of teratoma formation. This has already been demonstrated by various tests of the V cells. They don't do that. And in all of our experience, we haven't seen that come up. Yet they have the ability to become anything. And what they become is a function of the environment that they find themselves in. When I began this work, I was working with a specialist in the blood field, a hematologist, and he showed me that depending upon the chemical microenvironment of the V cell, that it could become a neuron or a muscle cell or a cartilage cell or a pancreatic islet cell that makes insulin. So in part, it's the chemical microenvironment that tells it what to become. And it's also about cell-to-cell communication. Cells have what are called cell adhesion molecules, a metaphoric Velcro of the cell that allows them to stick to each other. And the first class we looked at was integrins on a different type of stem cell from the bone marrow that expresses a marker called CD34, and this is a blood-forming type of cell. And they have a class of molecules called integrins because they maintain the integrity of the structure of cells as they adhere to each other to create an organ. It's also involved in cell-to-cell communication. The cells transduce signals from one to another this way. So between cell-to-cell communication and the chemical microenvironment, that's how a stem cell that gets into a compartment knows that it's supposed to become. Once you get past pluripotent, you have germ layer derived. So you have mesoderm, ectoderm, and endoderm. And for practical purposes, what's really widely used now are from the mesoderm. These are so-called mesenchymal stem cells, or MSCs. And these are quite popular. They can be derived from fat. Curiously, you wouldn't think fat has a lot of stem cells, but actually it does. To derive them from a person, it generally requires doing a type of mini liposuction. The cells are digested, and there are ways to concentrate the actual mesenchymal stem cells from that process. And these have the predisposition, because they are mesodermal, to make cells of that type of germ layer. So they're predisposed to make muscle, bone, ligaments, cartilage, tendons, and even fat. So if someone has a challenge with one of those types of tissues and they're young, the MSCs can work brilliantly. Someone tears a meniscus in their 20-year-old athlete, mesenchymal stem cells from their fat or also from their bone marrow can work quite well because they're designed to become this kind of tissue. Then there is some work with tissue-specific stem cells. Very interestingly, when I began looking into the stem cell world, virtually every tissue in the body has its own type of stem cell. There's a liver stem cell and a muscle stem cell. There are 3 different types of neural stem cells to make new nerves. And even though they're tissue-specific, there really hasn't been nearly as much work in tissue-specific cells, although that could well become an area that gets developed. So if the most common types of stem cells used are mesenchymal stem cells, it's probably the predominant. They can even come from umbilical cord blood. Then you have the challenge that unless it's your own, you have the issue of whether they will actually stay in the tissue if they're not a tissue match. That's a whole different set of questions. We won't talk too much about allogeneic stem cells. Mesenchymal stem cells typically have a diameter of 12 to 30 microns, which is really small. However, if they're given intravenously, the first place that they go is the lung. The average diameter of a pulmonary capillary is 6 microns. So it's just a physical thing. It's not that easy to get a 12 to 30 micron ball through a 6 micron tube. Some of them can squeeze through, but I've seen studies where typically 95% of the MSCs get trapped in the lung. And if the goal is to get them to the lung, then it's a good delivery system. But if the goal is to get it to some other tissue, especially one that isn't of mesodermal origin, may not be as good of a choice. The other issue with mesenchymal stem cells is that they undergo an aging process. And there are studies that show that the older a person is, the less potent the MSCs tend to be. So a 20-year-old that injures their cartilage may get a tremendous result, but if someone's 80, there may not be much of a result. In contrast, the V cells are only 1 to 2 microns. I can show you a slide where we have looked at this under a microscope in one of the research labs with which we work, and they're literally about 1/4 the diameter of a red blood cell. And it's not that difficult for a 1 to 2 micron ball to get through a 6 micron tube. So one advantage of the B cells is that if they're given intravenously for a systemic purpose, they can easily get through the pulmonary circulation and then pretty much anywhere throughout the body. And with neurologic improvements that we've seen, although we haven't done an experiment like give these to lab animals with tagged stem cells to prove that they cross the blood-brain barrier. At least the clinical results are suggestive that they can and do. The other thing that's extraordinary and really unknown why about the V cells is that they go into a type of metabolic hibernation at around the time that we're born. And because they're metabolically very inactive, they aren't dividing, or at least do so very slowly, which means that their telomeres aren't shortening or very little. And we've done other work that, as expected, the V cells remain very young with respect to the biology of the person. The chronologic age of a person can be in the older decades, and the V cells remain many decades younger. So, as a type of stem cell, it's small enough to get through the lungs and get anywhere. It remains much younger biologically than the biologic age of the person, which is typically roughly the chronologic age, unless they do some very good biooptimization work. And depending upon where they go and what they connect with cell adhesion, they can literally become anything. The fact that they are at the tip of the differentiation chain, we believe that they're not only going to assist with the replacement of adult cells with young cells, we believe they're actually reversing the biologic age of the various stem cell pools of the body. At least that's our theory, which accounts for the effects where we see reversals of biologic age. There was a famous study in longevity that was done by a colleague named Dr. Greg Fahey. You probably recall this work where he combined growth hormone with metformin and DHEA, and he gave it for a year and saw about a 3-year biological age reversal, which was really great. This was even associated with thymic regeneration. They did CAT scans that show that the thymus, which tends to peak at age 15 and get smaller from year to year after that, which is where we make T cells of the immune system, which are quite important for various types of immunity, and this was being regenerated, so that looked great. And then I saw a presentation where he showed the results after 5 years, and the biologic age reversal had regressed back to the chronological age. When we first looked at biologic age with the V cells, we found that if someone had had a single treatment anytime in the past 5 years, that they were usually about 3 years younger biologically than their birth date age. If they hadn't had one of these treatments in general, they were about as old or even a bit older biologically than their birth date age. There have been a few exceptions with some biohackers, and in general, though, people were literally about as old biologically as their chronological age. What was really interesting is that if they'd had 2 treatments, they were about 6 years younger, and with 4 treatments, we saw 12 years younger. So we were seeing roughly a 3-year biological age reversal per treatment cumulatively, which is super cool. And then we were able to do pre and post-treatment testing once we got baseline data and could do follow-ups, and we found that typically 2 to 3 months later, we were seeing about a 3-year biological age reversal effect. There was one thing that neutralized the effect, which was corticosteroids. So we had a martial artist who was getting lots of cortisone injections into joints because they were hurting a lot from what he was doing. And we saw he didn't have the reversal effect that we thought. Occasionally, people under really high stress, so their endogenous cortisol levels were high due to that, and they didn't get the effect either. And at times that they weren't stressed, that they did. So that's the one factor that we found. There's a new paper in the area that suggests that lithium may have an inhibitory effect. We have to explore that further. And other than that, those are the things we found that may make the effects not as robust as what we usually see. So, as a way to be in a younger biological body using the most accurate test of biological age at the DNA level, which is based on the work of Dr. Stephen Horvath of UCLA. And the first clock of this type is in his honor called the Horvath clock, which looks at something called DNA methylation. When we start with one cell and ultimately have hundreds of types of cells, the way the body differentiates one type of cell from another is it places little chemical groups called methyl groups, which is just a carbon with 3 hydrogens, and very specific DNA residues, specifically of the 4 bases, it's the cytosine residue. So these modified bases are called 5-methylcytosines. And it has a very precise pattern that is specific from one type of cell to another. And just like when you make a copy of a copy of a copy, later copies tend to be not quite as high resolution. And there's a characteristic way that the methylation pattern tends to become less accurate than where it was at the beginning. And Dr. Horvath created a map of how that pattern changes over time and associates with biological age. The other time clock of DNA that people are acquainted with in a much broader way is the so-called telomere length. And telomeres are the ends of chromosomes. It's a specific pattern of 6 DNA bases that repeat over and over. And when the cells divide as part of the process, the telomeres get a little bit shorter. And when they get to a certain length, typically half the length that we're born with, the cells stop dividing and they either disappear through apoptosis or they become senescent, which it's actually kind of worse if we have senescent cells because they make inflammatory factors around them that accelerate the age of other cells. So with this most accurate clock, we're seeing biological age results that are quite significant in terms of biological age reversal. So if someone can reverse their age 3 years, it's literally reliving the last 3 years biologically. And it's not just so-called anti-aging or slowing the clock, it's actually turning it back and having that time to live in a younger biological body. And our admonition is always, well, if you're going to have 3 years, now you can exert a little intention. You can alter diet, lifestyle, exercise, sleep patterns, do yoga, meditation, all sorts of practices to enhance your abilities. And then a 3-year cycle, you can become an enhanced version of yourself, and then you can do it again.
Well, this sounds wonderful. So, one of the first questions I have is that there are people who are out there who advertise as stem cell physicians and experts. And from until I met you, I saw that what they are doing is getting umbilical cord stem cells, which seemed reasonable, very useful stem cells. Obviously, there's an issue in that you're getting cells from another body, which means there could be an immune reaction, which I don't care for. But with this information out there about the V cells, why aren't these stem cell physicians using it? Why are you the odd man out? And what's going on here?
Well, you have early adopters and late adopters in anything.
Yes.
And part of the V cell story, and it makes it even more interesting, is that they've been really controversial. And they were only discovered in 2006. They were first discovered in mice, and shortly thereafter in humans, and pretty much every mammal studied so far has them. And there was a famous scientist that basically declared they don't even exist. And it's taken quite a while for the literature in the area to be pretty conclusive that, yeah, they really exist. I mean, I can show you V cells derived from somebody's peripheral blood. You can see them on a microscope. They do exist. And they have early stage protein markers on their surface with alphabet soup like CXCR4, SSCA4, OCT3AC4, and these are the particular markers we've used in flow cytometry to characterize the number of cells in peripheral blood and actually to show that the process that we use to awaken them shows an incredibly interesting phenomenon. We did a study in Manchester, England, with a colleague of mine named Dr. Peter Hollins, and his claim to fame is that he was part of the team that discovered and developed the area of IVF. And he was part of a team that was ultimately awarded a Nobel Prize in 2008 with the mentor of the group, Sir Robert Edwards. So he's definitely a first-class scientist. And we did an experiment where we used a new type of laser we've developed, and this is technical. We call it the song laser for a variety of reasons. In part, we're singing the song of the stem cells in light to be able to direct them, to increase the probability that stem cells go where you'd like them to go. And what was interesting is that the actual cell counts per cc of so-called platelet-rich plasma, or PRP, and probably most of your listeners know that PRP is just derived by taking a tube of freshly drawn blood, putting it in a special tube with a gel separation, putting in a centrifuge, and through the density of the cells, you pull out the red and white cells, and the better it is, the more you remove both the red and white cells. If you're going to use the PRP, say, to inject a joint or ligament, you really don't want the red cells and white cells there because they tend to be inflammatory. So we have a system that creates white cell depleted PRP. About 98 to 99% of the red cells are removed and about 95% of the white cells. So there's very little there except plasma and platelets. And about 2/3 of the blood in most people is plasma. And the platelets are small, so they don't get pulled out into the gel, and the V cells are about the same size as platelets. So the first thing we confirmed was that we were losing very few V cells with this particular tube that we validated, less than 1%, so it was a very efficient way. And the cell count in the plasma using flow cytometry was about 800,000 per cc, which was a pretty good amount. And then we did a test to look at the optimum duration of using the song laser and found that 1 minute started the effect, 2 minutes was greater than 1, 3 minutes a little better than 2, and 6 minutes the effect was reduced, and 9 minutes was like not doing it at all. So we had found that optimum time is really interesting. Like other effects in photobiomodulation, more isn't necessarily better. There's a sweet spot. And what was remarkable, because the flow cytometry was done immediately after the laser process, that the cell counts went from 800,000 per cc to about 2 million per cc. And we know that cells don't divide that rapidly. And for those that like physics, we wrote a theoretical biophysics paper with the theory that we had used just the right type of laser energy and the right type of frequency patterning, that we, through resonance, dissociated a protein called EpiX4 from a V cell marker called CXCR4. And our theory is that we weren't doubling their numbers immediately because they were dividing. We simply removed a protein that was blocking the ability of the antibodies used in flow cytometry to bind them, because that's how they're counted, because the antibodies are bound to something fluorescent, and the intensity of fluorescence is how they estimate the counts. So we believe we were increasing the countability. And whether...
So you were saying then that by your processes, removing a protein from the blood sample allowed more cells to be counted. Is that correct?
Right. We believe it increased the countability. We don't believe the number increased because they could divide that fast.
Got it.
Now, whether that's part of the mechanism that awakens the cells from the metabolic dormancy, that's another basic science question that will take some more work to prove one way or another. It may be that mechanism or others, or some combination of mechanisms. What's also interesting is that when the cells are in their dormant state, they are about the size of bare nuclei. A nuclear diameter is about 1 micron typically. So these were cells that essentially looked like bare nuclei with a little bit of cytoplasm or hardly any detectable cytoplasm. And 4 days after this stimulation process, they'd expanded their cytoplasm to be 2 to 3 times the diameter. And they were also starting to create what are called colony forming units. So the cells were able to expand their numbers as well.
I'm taking notes because I'm going to have to write about this to entice people to watch this. So through this process, you're saying that you were able, in your belief, to get more countable of these stem cells, V cells,
Mhm.
very small embryonic cells, correct?
Mhm. Very small embryonic-like, VSCL.
Embryonic-like cells. Okay. And not only that, you were able to, this process also turned them on to become awakened.
Right. And
Okay.
that has to do with what we're doing with the laser itself. And song really has two meanings. We are singing the song of the stem cells in light. Our very first experiment wasn't done with B cells, it was done with a type of stem cell from the bone marrow. And this was before we knew it would do anything. We created a new type of laser waveform that, in theory, would have much greater depth of penetration, but might not interact with anything. So it might be kind of a cool physics experiment, but not be useful. So a colleague of mine at the NIH gave me 5 flasks of a stem cell-like cell from the bone marrow. She was in bone marrow transplantation. And like a good experiment, we did a dose-response curve. So in the first flask, we applied the laser for 5 minutes, then 10, 15, 30, and 60 minutes. We applied the beam along the long axis of a typical size culture flask, put them in the incubator, and then hope that one day later we see something interesting. It was one of those scenarios where you literally like throw it against the wall and you hope that there's some amplification cascades in the cells that will show you that it actually made a difference that might be meaningful or useful. And what we saw in every single flask from 5 minutes to 60 minutes was a column of cells where there were so many cells you could literally see it with the naked eye where the beam had been. So we had somehow stumbled upon or fortuitously came upon how to sing the song of the stem cells in light. And that particular type of cell uses a system called the integrins. And integrin molecules are a dimer. There's one alpha chain and one beta chain. And it's a really cool protein because it has the extramembranous part, which is the little Velcro hooks that allow the cells to stick together. There's an intramembranous part, which may have a trigger zone. And then there's an intracellular part, which associates with transduction proteins. So the integrins are also involved in cell-to-cell communication and transducing information from one cell to another. And what was interesting, because we're trying to figure out, well, why are these cells sticking to each other? Because that column remained coherent unless you could shake it up. The cells would break apart if you agitated it. but they were sticking together. So the question was, are we altering cell adhesion? And what's interesting is that we found that 24 hours after a single stimulation, that the physical expression of beta and alpha integrins increased as much as 40 to 100%. So we're literally upregulating the ability of the cells to stick to each other and by extension probably to talk to each other. The more sciencey aspect of what we're doing with the laser is the creation of an optical device, and this was a collaborative project with me and a super smart physicist from Edinburgh, Scotland, named Scott Strachan. Love the name Strachan. And he's a big fan of William Wallace and drinking good brew in the pub. The typical laser, the usual lasers that we're accustomed with, create waves of the same wavelength that are in phase.
Yeah.
And if you add waves in phase, it's literally just algebra. The sum is an amplified signal. And because of things like entanglement and the fact that the waves are in phase, you get the phenomenon of coherence, which is why if you hold a laser pointer against the wall and pull it away, it stays about the same size. If you hold a flashlight, when you walk away, the beam gets wider. It's coherent light versus incoherent light. The art of what Scott created for me was an optical device where we could align the waves exactly out of phase. And if you do 180-degree phase shift of two sine waves, you can even do it on a calculator in this graphics, and combine them together, you get a straight line.
Right. It should be zero. Should they not cancel each other out?
They do. You get a vector sum zero.
Okay.
And because there's no net electric or magnetic field, and this is how Scott modeled it, it will not interact in the same way with valence electrons because they interact with the electric and magnetic field of the photon. If it interacts, and this is another theoretical physicist's feeling about it, the interaction would be with the nucleus, which is modeled as a high point density, a so-called nonlinearity in the vacuum of space. Some physicists even model it as a type of mini black hole, which could deconjugate the signal. And there's some other interesting aspects even of an equivalent waveform where the waves are in phase but going in opposite directions. And this is where it gets kind of cool science fiction-like and a bit provocative, is that it's still a vector sum zero, but it's waves going in opposite directions. And even conventional physics would hold that the wave going to the right is not just going forward in space, but also going forward in time. And the wave going in the other direction is not only going backwards in space, but backwards in time. And that gets into concepts of timelessness, instantaneity of communication, and really cool stuff like that. And if we say that the interaction is with the nucleus versus the outer electrons in the valence shells, then we talk about the relative diameters. So an atom is really small. The model of the hydrogen atom is that the electrons are in a spherical shell about a diameter of 1 angstrom, and the radius would be half of that. So if the diameter is 1 angstrom, that's really small. It's 10 to the tiniest 10th meters, or it's basically a tenth of a nanometer. However, the nuclear radius is modeled to be 10 to the minus 15
Right.
meters. So the ratio is that the outer shells are about 100,000 times the diameter. With that relationship, if the interaction is the nucleus or a short distance around the nucleus, in theory, the vector sum zero wave with no net E or B field would go as much as 100,000 times deeper through a medium, which then gets into a depth of penetration question. And Scott modeled that the residual of light, because we can actually adjust the percentage of the photons that go into this wave cancellation or this vector sum zero, where the waves are added exactly out of phase. And the residual of light is a different pattern than the laser itself, because you get into a combined destructive and constructive interference pattern. And just to describe it, it would be, instead of a continuous beam of light, it's mostly dark with little islands of light sparkling. And his modeling suggests that even the physical light, the physical photons, would be able to penetrate 10 to 20 times deeper through tissue than ordinary laser light. And his theory has to do with reduction of scatter pathways, because when an electron absorbs a photon, it becomes reflective of the next photon. And even getting physical light deeper into tissue can have many photobiomodulation benefits if you get past the surface. The ability of the beam to penetrate then suggested that we can increase the probability of a stem cell going where you want it to go if you provide a signal that attracts the stem cells to where you want them to go and makes them stickier. So step one of the process is awakening the cells. These cells are other, actually. And then the second part, when we fuse them IV, they can go anywhere. And then we apply the laser with the intention of increasing the probability that more stem cells will go in the part of the body where the person could most use a regenerative stimulus. So song.
That kind of blew me away. And you might remember I said to you that it sounded to me like the image of the duckling. The duckling comes out of the egg, and it's going to fixate on the first image it sees, which is generally its mother.
Mm-hmm.
And then it's going to follow its mom.
Mm-hmm.
But if it sees you first, it's going to follow you.
Mm-hmm.
So the metaphor, the analogy I had is you're training these cells with light. They see the light, or they experience the light, or somehow they can see it.
Mm-hmm.
And that tells them, oh, I'm attracted to you, Mr. Frequency. And then you administer that frequency to where you want the cells to go, and they circulate. And then when they experience that frequency in the tissue you're beaming, they nestle there. Is that accurate?
That's what we believe. Mm-hmm.
Okay.
Good description.
I'll collect my fee later. I've been writing for years, and I really try to write in a fashion that the layman's going to understand. So the cells initially, you give them the light initially. That sets them up, it awakens them, and it programs them to want more of this light. So now they're awakened. You administer the cells intravenously. They can get anywhere because they're smaller than pulmonary capillaries. And when they experience that frequency and whatever tissue they're going through, if you're beaming that light on that tissue, they're just going to come to a full stop right there and start adhering. Is that it?
I think the likeliest mechanism of the increased probability of the cells going where we want them to go is increased cell adhesion. They're just sticky. They get there and they stick there.
Okay. And that's because they're experiencing that frequency there.
Mm-hmm.
And that makes them probably get sticky right on the spot.
Mm-hmm.
Okay. So, what I'm hearing from you, forgive me while I take a note. What I'm hearing from you is your system enables you to get a hell of a lot of even more primitive stem cells that can go in virtually any direction. They're not limited to mesenchyme. They're not limited to endoderm or ectoderm. And for the viewer, as you said, mesoderm is muscle, connective tissue, cartilage, even fat. Ectoderm in embryology moves on to skin and neural tissue. And endoderm would be lungs, gut, liver, some internal organs.
Mm-hmm.
So these cells can go in any direction, not just one, and you then can direct them to the location you want them to go.
Mm-hmm.
And it sounds like a lot of this was found serendipitously, or was it deliberate? Or was it God?
Well, it's probably a combination of fortunate inspiration and observation, a lot of serendipity, a lot of luck along the way.
Okay. I think this was all said in a fashion that the average viewer can understand, except for the stuff I couldn't understand either. You know, those proteins and all the letters for molecules and such. So this could be a major step forward. And now I would like you to tell the viewer what you showed Terry and me and the group at this meeting that made our jaws drop.
Which part of it?
Well, let's start with the neurological, because the neurological would be hard for almost anybody to believe, and yet you've presented the unbelievable. Let's start there.
Well, first a disclaimer that we're not saying that we we treat anything.
Of course. Well, let's say you're not treating anything, you're attempting to restore health.
Right, it's regenerative.
Yes.
So if the goal is regenerative and the cells are young and they can go and support a system, we can use Parkinson's for an example. And it's a good test of this particular theory because we know that Parkinson's is caused by a dysfunction in a very precisely located part of the brain called the substantia nigra, which means black substance. And it's actually physically darker if you look at a physical brain. And it's because the high presence of dopamine will give it that color. And it's depletion of the dopamine producing neurons that results in the movement disorder because it's required for integration of coordinated body movement. And when we address it, we have the intention of giving the strongest signal for the cells to find their new home in that very precisely located part of the brain. It's about a marble-sized nucleus that rests close to the floor of the skull, roughly on a line if you drew a line right above the tip of your ear, and coming down just about 1.5 centimeters lateral to the midline. So for that, we direct this signal at that particular part of the brain from 3 different directions. So we can maximize the signal. The signal will be all the way along the path, but if we cross the signal at that particular location, it's sort of a summation. If you're doing math, it's like the integral of where the signal is in a summation throughout the tissue. And what's interesting is that in many cases, even within the first hour, the regenerative signal has resulted in improvement in the movement disorder. So we have lots of pre and post videos that there is a visible difference even that quickly.
You explained that to me earlier. Please explain that to the viewer because it would be seeing the time it takes for neurological injuries to heal. It's fantastically in the next millennia that I would expect to see a result like this. Explain why it might work so quickly.
Mm-hmm. Well, I will actually give the explanation of a Harvard neuroscientist. He was a really good example of a fairly extreme situation. And he had a diving injury, really quite bizarre. He was 300 feet under the ocean.
300.
300. Yeah. Roughly 10 atmospheres of pressure.
Yeah.
And he probably got a little nitrogen bubble in his spinal artery because at that depth he became quadriplegic.
Oh.
And they brought him to the surface and did a very, as was described to him, high-risk hyperbaric oxygen procedure, which recovered a lot of function, but not all of it. And for 35 years, he had various neurologic issues, one of which was what he described as neurogenic numbness in his left leg. And we did the procedure for him, and in just 3 hours, he said his sensation was essentially the same as the other leg. And he said, our neurologic model cannot account for how this is happening so quickly.
Mm-hmm.
And then he gave a theory, and I think this is the best theory to date, which is that there can be nerves that are called idling nerves. They're still alive, but don't have enough juice to give the signals that they do to other nerves. And if there are potent cytokines and growth factors that quickly give the nerve more juice, that they can functionalize and start connecting and communicating. And that's called the paracrine effect, where cells that are releasing a chemical in their vicinity can have an effect on other cells. So these rapid changes that are really much faster than we expect for true new nerve formation or neurogenesis, we ascribe to this so-called paracrine effect for idling nerves, are brought in line like your computer is idling and you hit the button and everything comes up, something like that. And then he described another phenomenon with the palms of his hands, where his palms are really numb. And he said he would reach into his pocket and he couldn't tell a key from a coin. If he held a paper cup, he would crush it because he didn't have the feedback of how hard he was pressing. And that came back very gradually over several weeks until it was, he said, about 90% of normal. And that time frame he felt was consistent with neurogenesis.
Thank you. Now I'm going to play devil's advocate.
All right. I'm on the spot.
Okay. There's another therapy out there that a lot of doctors do called exosomes.
Mhm.
They are supposedly extracted from stem cells or factors.
Mhm.
And here you're talking about factors that can stimulate growth.
Mhm.
So the exosomes are allegedly, now I don't do either of these.
Mhm.
I'm very interested in what you do. So if I'm going to go in a direction, I'll probably go there. But exosomes are said to be far, far less expensive and have good effects because of these factors.
Mhm.
What's going to be the difference between that and what you just talked about?
That is such a good question.
Oh, that's what I'm supposed to be doing.
Well, it's really pretty straightforward. All an exosome is when you define how they're created, is a little bit of cytoplasm from the cell that forms into a little vesicle that contains cellular content. It'll have cytokines and growth factors, whatever that cell happens to be making at the time. And they're very small, which has some utility. And there are a couple of fundamental issues in this whole area of exosomes. And one of my colleagues reviewed this quite beautifully at a recent event. The first issue is that these are a byproduct of living cells in culture. Living cells in culture don't remain the same. They evolve over time. And unless a particular exosome company is doing rigorous and strict quality control, and they're culturing these cells over time, I have witnessed what I would call a cell passage effect. And in the earliest days, especially when I did some work in other countries, I was working with cord blood stem cells. And I did a clinical study and did a dozen treatments, and they gave a really nice result. No significant adverse effects and really good clinical effects. Then from the same company, I got another dozen sets of cells, and 2/3 of people had an intense inflammatory reaction, which was like getting the flu.
Now, were this cells or exosomes you were getting?
These were cells, but I'm making a point about passage.
Got it.
So I said, like, what's going on? This batch of cell units did great. And this other batch was inflammatory. We didn't get the regeneration effects we were looking for. And people had this inflammatory reaction like the flu with feverishness and achiness and arthralgias, joint and muscle discomfort. And I had to press them. But what I found out was that there was a difference in the passage number. And a passage is this. One of the issues with MSCs is that in certain jurisdictions, you're allowed to expand them. And the way you expand them, you get the fresh sample, say it's cord blood, you get the MSCs, and you put them in a culture flask. And you give them food, and you put them in an incubator, and they multiply. And they multiply to confluence, that there's this carpet of these cells. And when they expand to confluence, then you would say take those cells and put them in 10 other flasks. And that would be passage 1. You started at passage 0, they haven't a passage yet, and now it's in the first passage. And then you grow all those flasks to confluence, and you put each of those in 10 flasks, and that's passage number 2. And the passages continue that way. And what I learned was that the cells that worked well were low passage. They weren't more than 3 to 5 passages. The cells that were inflammatory were 7 to 9 passages. So as the passage number increased, it went from regenerative to more inflammatory. So an issue with exosomes is, well, are these low passage number exosomes? Because they're literally just little bits of cytoplasm that have broken off from the cell membrane to make a little vesicle, or are they high passage? And if there isn't really strict control from one batch to another, just like my experience, one batch could be really great and regenerative, and the next batch could give a more inflammatory result.
Yeah, that's frightening. Lucas, I hope you had the look on my face. Keep that in there, because I thank you for that. That is really, really frightening. Same company, same cell line, but the more they propagated the cells, which makes them more money because they have more cells, the more inflammatory the response.
That's what we saw, and that would be the quality control question I would ask of any company providing exosomes is what's the QC, what's the passage number, and how do we know one batch is going to be very comparable to the next batch? So if you get a good result with one, that you can expect a good result with the next.
If you could guarantee that you were getting a low passage and you knew it, would you expect to see halfway reasonable results that could vie with what you're doing with your V cells?
The next question in this area, with the first, I think, being the most important question. How do you know that if you order the same thing, you're really getting the same thing? Because it's not a biochemical product. These are the products of living cells that are dynamic. The second issue is that exosomes can have very powerful growth factors and cytokines, and that can be beneficial, depending upon what they're being used for, whether that set of cytokines and growth factors is a good fit for the application that you're using. What you're giving is basically, with a single exosome treatment, is a single shot of cytokines and growth factors. It's kind of like PRP. It's a single shot of cytokines and growth factors, and that can be beneficial. But you're not actually doing a cellular process where you're giving cells that can be specifically regenerative. And I did the math on it. There was a case where the person got 15 billion exosomes with an average diameter of 130 nanometers. And then I estimated with the V cells, based upon our flow cytometry data, that the V cell treatment was about 50 million cells. If you consider the source of exosomes being the cytoplasm, then a comparison would be, what's the total cytoplasmic volume in an exosome treatment versus a typical V cell treatment? And 15 billion sounds like a lot more than 50 million, but the exosomes are much smaller. So I did a volumetric calculation, because the V cells will be about 5 microns in a few days. And the math suggested that the single V cell treatment was giving roughly 100 times the cytokine content of the exosome treatment that was given. And it's actually pluripotent stem cells that can replace and regenerate cells in tissue. So it's doing more than that. And the V cells make exosomes. Little bits of their cytoplasm break off. There'll be an ongoing source of exosomes.
So the V cells then remain alive in the tissue, and they can create more growth factors. And even if they split off their cytoplasm as exosomes, they're going to regenerate and make more, so it's ongoing. Exactly. It's not a single shot.
Exactly.
Okay. Well, I like that. So give us a few more examples. You've told us about a neurologic injury. I'll share with the viewer briefly that I saw a video of a Mexican officer shot in the spine. Was the spinal cord transected?
It was not. That's really important. That's one of the questions, whether it's a ligament like the anterior cruciate ligament, the ACL in the knee, or a peripheral nerve injury or a spinal cord injury. If it's transected, then the ends are separated, and you're not going to be able to bridge it unless you can bring the ends together, and that's typically going to be surgical. So in this case, it was more a spinal cord
Shock.
contusion, spinal cord shock that caused a complete spinal cord injury, meaning that there was no sensation or motor function below the level of the injury. But the spinal cord was still continuous, so there was a physical structure that could be bridged across.
Okay. And that did take a couple weeks for him to recover.
It was actually more than that. We did a series of 3 treatments, and it was about 6 weeks after the first treatment that his sensory level improved from the mid-chest around 3 or 4 spinal levels down. So he had sensation much lower than he had before. It was also associated with recovery of erectile function, which he was quite pleased about.
Yeah.
And that's where it started. And we often find that there is a stepwise approach where you regenerate a system one step after another. After the second treatment, again, ballpark 6 to 8 weeks later, he had the experience where his sister was cutting his toenails and he said, "Ouch." And she said, "You can't feel that." I said, "Yes, I can." So his sensation returned all the way down to his toes, and he actually had the ability to move his toes a little bit. So that was a big step in terms of a connection with at least some function all the way down to his feet. And the third treatment that we did, which we did at a clinic in Mexico, where we're allowed to do this, was actually an intrathecal procedure where
Wow.
the stem cells were actually given directly into the spinal fluid through a lumbar approach with an anesthesiologist that did a lot of that. And that and several weeks of rehab, then he actually regained the ability to walk and ride a bike again.
That's phenomenal.
Before we jump back into our discussion, I want to remind you about the amazing health benefits of ozone therapy with Simply O3's home systems. Imagine feeling increased well-being, sharper mental clarity, and less inflammation after just one treatment. The Simply O3 3.0 Complete Kit lets you experience these benefits in the comfort of your own home. It's equipped with laboratory-grade materials to ensure safety and efficacy. This kit offers thousands of treatments and is far more cost-effective than clinic visits. Don't forget to use the code in the description at simplyo3.com for 10% off. I'd also like to give a quick shout out to PROMOLIFE. If you haven't heard of them, they're leading the game in ozone therapy. Crafted with care right here in the USA, their ozone generators are top notch. Beyond that, they've got a full lineup of accessories that truly opens up the world of ozone in innovative ways. To support this podcast and check out what they have to offer, you can visit them at www.promolife.com/ozone. And as a little perk for you, our listeners, there's an exclusive discount code waiting for you in the podcast description below.
Now, for the viewer, I also saw videos of Parkinson's patients who could barely get out of a chair, and shortly after your treatment, they were dancing. Is that accurate?
That's true. That is
Okay. I mean, this was humbling to see. And like I said, Terry and I were astounded. So we've seen, or at least you've told us about Parkinson's, a spinal cord injury. What about MS or ALS?
Well, MS is really 2 issues, which is an autoimmune inflammatory process of the myelin sheaths of nerves. And because the target is myelin, it can create insults almost anywhere throughout the central nervous system. So what's challenging and typical is that if you look at the scans of the brain and spinal cord, there can be lesions that occur almost anywhere. There can be a few of them, or there can be a lot of
Right.
them. It can be all through the brain and
So
spinal.
you have a problem there in focusing where you want the cells to go.
Well, in that case, we direct them to the entire brain and spinal cord.
Okay.
That's where we develop what we call the whole brain procedure. Since we know it can be anywhere, not like a very localized condition like Parkinson's, where we know exactly where to focus. If it can go anywhere, then we send them generally throughout the central nervous system.
Do you see results then with MS?
Well, okay, you're pushing me. These are just regenerative effects.
I understand.
Just regenerating the system, and things can improve if you regenerate. The most dramatic case was a person who was in active relapse of MS, and as you know, there are different flavors of MS,
Yes.
and particularly common is relapsing remitting, where there's an acute aggravation with different neurologic insults and challenges, and then that cools off and they get better, but often they don't recover everything, and
Right.
then they're fine for a while and have another exacerbation. So she was in the midst of a flare, and she was having trouble speaking, chewing, and swallowing, and had weakness on the left side of her body. Within an hour after the procedure, she said she felt stronger than she had in 8 years, and she put an exclamation point on it by running a full marathon 2 weeks later.
And this is one treatment.
Mm-hmm.
6 weeks later.
No, it was 2 weeks later.
Two weeks later.
Yeah.
Even Lucas is smiling at that one. Okay. So that's MS. What about ALS, which could be also broad?
Mm-hmm. So-called motor neuron disease.
Yeah, the motor neurons for the layperson, they're dying in ALS, Lou Gehrig's disease. You're doing regenerative therapy. You're not treating Lou Gehrig's disease. We'll grant you that. Can this therapy possibly regenerate some of the motor neurons?
We did a treatment for someone, and I won't go into the specifics of protocols and how we stack things, all the minutiae of that. We often find we need to be more aggressive with conditions like that because there's so much neuronal injury that needs to be overcome. And then if the motor nerves are not working well, then you also complicate it with muscle atrophy because they're not exercising properly either. And this was a case from Israel, and we did a more aggressive protocol. Sometimes we even draw more blood and basically increase the number of cells we have to work with. And he wasn't able to move his right arm for a year, and after the treatment, he could move it again.
Okay. How did he end up doing at the end?
Well, the issue with neurodegenerative conditions is that the most likely, or at least the most strongly postulated process is a protein misfold issue,
Okay.
a so-called proteinopathy. And there is a typical course of how the nervous system has a progression of that issue. It's a lot slower in some conditions, a lot faster in other. There's a range of what's typically seen, and ALS is one of the more aggressive ones. So with the regenerative approach, we can see significant improvement, and then the underlying proteinopathy takes over, and
Yeah.
it will tend to progress.
Okay.
And then we need to treat again to get a stabilization or improvement. And so it's more of a staircase thing where, depending upon what it is, the number of treatments may be shorter or longer in between to at least create the sum of a stabilization and a better course over time than without having periods of getting better.
Okay. Let's move from the nervous system to something very common. There's an epidemic of macular degeneration today, and I have it. You know that. My vision is still good. I've been blessed. Um, have you tried that?
Mm. Yeah.
What might, can you get regeneration of the retinal pigment epithelium with stem cells?
Eye issues are really interesting. And there's a couple of genetic conditions that we've even improved with that issue. One that we may study in fine detail with the most advanced retinal testing methods. What we can say functionally, again, with regeneration of the system, and when we address the eyes, we really address the whole system. So it wouldn't just be the retina or even just the eye. We would address the optic nerve, the optical radiations through the cortex,
Yeah.
and ultimately the occipital cortex, where the information is processed. And we have a number of cases where, again, surprisingly, within the hour, that people are able to see 2 and even as much as 4 lines better down the eye chart. The most dramatic was someone with macular degeneration. This is purely regenerative, who could only see part of the big E on the top of the eye chart, and in 45 minutes, she could see 4 lines down the eye chart, in what she described as film or filters over her vision had substantially cleared.
You're giving me goosebumps when I hear that. How about more common things like organ failure, kidney failure, where people need dialysis? Can you regenerate renal cells
That
or heart cells in heart failure, failing organs? Can this help that?
we published a study from Armenia. It's a pilot study, only 10 people, but with very dramatic results. And to review heart failure for the listener, to have an understanding of the significance of these numbers, a normal heart pumps out about 50 to 65% of the blood with each beat. That percentage of blood is called the ejection fraction.
Right.
If it drops below 50%, it's considered mild heart failure. Below 40% is moderate heart failure. Below 30% is severe heart failure. That puts it in a context. This clinical study was all persons with severe end-stage heart failure due to the most common causes, which is ischemic heart disease and hypertension. And this was under the auspices of the Armenian government that was supportive in a hospital in the capital Yerevan in the Department of Cardiology. And the metric we're following was the standard accepted one, which is echocardiograms.
Yes.
And then at a high level, all the echoes were done by actual cardiologists, not by technicians, which arguably is good for accuracy. The average ejection fraction of the group was only 21%. They were essentially heart transplant candidates.
Yes.
And the comparison was a so-called meta-analysis, where multiple published studies are summated, and this was 1,494 cases of using intracoronary stem cells. So delivery method was actually doing a cardiac cath, putting a tube in a coronary artery, and injecting the stem cells directly into the coronary blood supply. So as
this is standard stem cells, not your stem cells, correct?
right, this was autologous stem cells from the person that were delivered by intracoronary injection. The average result 6 months later was about an 8% improvement in function, which was better than they started. In contrast, because of a case that I had seen where a person whose ejection fraction was 17% was clinically better within a few hours, and because I actually was physically in the other country, we brought everybody back for another echo only 3 days after the treatment. And the average improvement in 3 days was 14%. And that was already statistically significant. Then at the 1 month point, it was on average 25% better.
Wow.
At 2 months, it was 37%. And then at 3 months, it plateaued at a 50% average improvement. And the results were such that about half of the subjects improved off the transplant list, crossing a threshold of 30%, which has a better prognosis, and 20% improved almost into the normal range with a single treatment. And this was intravenous with laser guidance, not intracoronary. The survival statistics were also relevant. The prognosis for someone with an ejection fraction less than 30% is only about a 30% 1-year survival. And we saw an 80% 2-year survival, where that was as long as the follow-up went. And what was relevant was that the only 2 people that didn't make it were the people on the low end of the response. And this is another really important point. Whereas the average of the big meta-analysis was an 8% improvement, and the average of this clinical trial was a 50% improvement, there was a really big range. It was a 15% improvement on the low end, which was still significant, or at least better numerically than the other, but the high end was 115%. So there was a huge range of response, even though these were very well stratified, similar age, similar cause of the issue. There's this whole unknown about why there's so much individual variation in response, from 15% to 115%. And the only 2 people that didn't make it were on the low end with a 15% response. This was a single treatment. We know from other work that if we do additional treatments, we can step it up. I think they only went from 20 to 23%, but with another treatment, it could have gone to 25 or 26, although they might have crossed the 30% threshold. So
how many were in your group?
That was 10 subjects.
And okay.
And this is actually published, so I can send you the full text publication.
With this kind of stuff out there and it's published, why aren't cardiologists doing it?
Awareness, probably.
Even though it costs thousands of dollars,
Mhm.
it's still a lot cheaper than what these people are going through.
Well, the cost of a heart transplant is about $1 million.
Yeah.
The alternative of ventricular assist device, last time I looked at it, was about $350,000. So, yeah.
There's certain things I don't understand. Let's move from heart, say, to kidney failure, which is common. We've seen a fair number of patients, say, with creatinines of 4 or 5, and ozone actually helps most of them. Not all of them, but most of them.
Mhm.
Have you tried it on stage 3, 4 kidney failure? Regenerative therapy to regenerate kidney cells.
Mhm. The kidney issue is interesting because if someone is in a state of milder renal insufficiency, creatinine above 1.5, but not going into the extreme zone where they're in the dialysis requirement range, we haven't really taken on end-stage kidney failure on dialysis, primarily because at that point the kidneys are so damaged and scarred that there literally may not be a place for the cells to go and regenerate tissue. With lesser degrees, where there is an architecture where the cells can go, a place that they can provide replacement cells and more functional cells, we have seen creatinine levels improve. Probably the most dramatic was one of the treatments done in Puerto Rico. And I am providing this technology under license to a number of really great clinicians in different specialties around the country. And we do have one cardiologist who's starting to generate data from us, has a very interesting case that we may publish actually. And that case was a diabetic with a creatinine of 6 that improved to 3. So
that's really, really good.
Yeah.
Because a creatinine of 6 is basically end-stage renal failure.
hmm
And 3, that patient could live years.
Right.
All right, what about common things like injuries, like lumbar disc disease, arthritis, etc.? Can you direct the stem cells to joints and have cartilage regenerate and see improvement?
I'd say what we do the most of is neurologic, cardiac, and joint if we are regenerating particular systems. Although our most widely used procedure is simple anti-aging, where we really believe we're going into biologic age reversal. So most of we see healthy people, they just want to be biologically younger. Yet we do lots of musculoskeletal issues. And there again, it's regenerative, and when you regenerate the tissue, it improves function and symptoms get better. So we've seen a lot of benefits with knees and hips and shoulders and low backs.
I got another question for you. We've been on a long time, and I really, really appreciate this. How does your laser penetrate the skull to direct the cells to the substantia nigra?
Well, that was the principle I was describing before, which is when we combine the waves out of phase and create a vector sum 0, our belief is that signal is going all the way through the brain from one side to the other. We had a very interesting experience where, with our latest generation technology, where we refined the optical relationships even further, where the person that experienced the previous generation and this new generation, they felt even more strongly. And then when we passed it through the brain of the friend they were with, it was going through that person's brain, and they felt it in their brain too. But they felt it differently, as though the brainwave patterns of that person being modulated onto the beam and being transmitted into hers.
Oh my God. We're getting into the stuff of movies now.
Yeah, kind of. I
All
love
right.
sci-fi.
Is there anything that I didn't ask you that you feel is necessary to share with our audience?
The one other point that I would make, other than understanding that there are multiple different modalities in this regenerative world, different types of stem cells, exosomes can be a factor, but as I said, it's really good to question the source about the QC aspects of them to make sure that there is batch-to-batch similarity of what you're getting, that it's good to have an understanding of whether the type, source, preparation method, and delivery method is appropriate for a circumstance that at least has a reasonable likelihood of giving a beneficial result. And the other thing we haven't really talked about is a phrase I've created called the biohack is the stack. That doing the regenerative medicine part with autologous pluripotent very young stem cells works better if the biological terrain is improved.
Of course.
Metals are cleared, if mycotoxins are addressed, if biofilm is addressed. A lot of what you do with ozone therapy is really taking care of those things so the landscape is better, and that alone can create a lot of benefits. So there's the biological terrain part of the stack, and there's all the things that you do after to bio-optimize the results. So there's nutritional metabolic approaches, and we've created a variety of formulas that we've clinically validated for that are also laser enhanced. And there are many other supplements besides what we produce that really can target a system to improve the biochemical pathways and give the cells what they need to regenerate. We work a lot with peptide therapy, and I won't say much about that. Those can be used quite constructively. That could be a whole discussion unto itself. There are other modalities, like the ones that you're doing. There's cryotherapy, infrared sauna therapy. There's what you're doing with ozone. There's pulsed EMF therapy. There's low-level laser therapy that have different types of photobiomodulation benefits from what we're using, but there can also be effects and a wide range of other tech. So the biooptimization is really what I consider almost a symphony, that it's what you're using with what intensity, how you evolve the theme over time, how you get to a crescendo, how you get to a resolution, and then what's the next round of the subsequent symphony.
Makes good sense, and I like the term symphony. I like harmony. You need several instruments to make a good orchestra. Lucas, you're a layman. I've seen your expressions in this. You're on now, like you are in all of the podcasts. Comments, questions for him, for the layman?
Yeah, actually, I just wanted to. So I don't want to get too far off topic, and maybe we need to have another podcast to get more into detail on this. But you talked about the symphony, the frequencies. I want to know. You talked about pineal tones and like the power of humming. I want to know if and how that relates to this sort of regenerative frequency therapy.
That's a beautiful question. And in a whole different domain of aesthetics and intuition and more the realm of the esoteric, yet that also has its own set of powerful effects. And I'm trying to figure out how to summarize it in a way that, with the time that we have, I can at least make sense of it. Around once a year, I gather what I would consider to be a type of interdimensional choir. And this is a whole system that I've been inspired to create. Some say I remember it from my soul history. A series of ancient tones that are sung together in pairs with large groups of people. And we've had as many as 900 people from over 60 countries gathering. And when these tone patterns, which each have an intention, they're done in pairs, are done in an order and sequence, it can create profound physical benefits. We've seen physical conditions melt away over the 3 days, where it's 2 days of preparation with everybody training together with our co-conductors, and then the actual choir presentation itself, which is more focused on expanding consciousness than specifically physical health. And many health conditions have improved or disappeared just by being in that energy vibrational environment. And then there are awareness effects that get more into the realm of our supernormal abilities that can unfold and expand with it. They're probably more of its own discussion, because there are now 36 different levels of tones that have evolved over the last 12 years. And it can create profound remembrances of who you are, what you've been, why you're here, where you can go, how you connect to your own higher information. It's a different realm beyond where we usually are in the technical aspects of medicine. And for those who may be interested in that, we have our 12th choir of 12, the final choir in the series, called the Aloha Choir, poetically on 12/12/24. And we expect over 1,000 choristers to gather on the island of Maui, where it said it was last done a galactic cycle ago. Is that a good enough summary?
Lucas?
That's perfect. That's yeah. I didn't know if we were going to be able to get into the details of this, because obviously there's a lot more there than a 3-minute summary.
Mm-hmm.
Now, this question I had for you: what exactly does gematria mean? What does that word mean? What is your use of that?
Great question. You do your esoteric homework. So, gematria is the name of the laser-enhanced nutritional supplement company that actually was the first company I created that supported the research up to this point. And there are 30 different formulas, 12 of which have been subjected to controlled clinical trials with very good results. A number of them have been published, one even patented. And the origin of the word is very ancient. It goes back to the mystery schools, especially associated with Kabbalah, where gematria, the way it was pronounced then, was the process of taking the letters in words and translating them into the numbers associated with them, and the deeper knowledge was coded in the pattern of the numbers. That's how the mystery schools actually preserved and transmitted the even deeper layers of esoteric information. I first saw the term, though, in a book you might have read called The Keys of Enoch, the Book of Knowledge by J.J. Hurtak. And in that book, gematria is defined as a science of determining the input energy required to build a body of consciousness, which is also something we're quite interested in. And then there's the most recent definition, which is gematria, are higher dimensional waveform representations that interact in matrices to create physical reality.
Lucas, one day we may have to have a podcast on the non-physical aspects of our existence.
I can get behind that 100%.
I'm sure you could. It could be a turnoff to a lot of people. It could be a real turn on to a lot of people. You know, we've been on for an hour and a half now, I think. I saw international news reports in the last couple of weeks on scientists taking a hard look at death, real conventional scientists, and they're questioning everything we know about what we are, who we are, what death is, and what remains after the brain stops functioning. So I don't think we're ever going to get to it all because this moves into the infinite. And while we're part of the infinite, we're not infinite. At least from my perspective, I believe we're part of the infinite. Would you agree with that, Todd?
Oh, this is a very deep discussion that probably warrants more than a sentence or two.
Yeah, we can't. We probably can't do it here. We can't. I liken it to the concept that the universe is a hologram, and you can cut the hologram in half and have the same information, and you keep going down to just a tiny part and shine a beam through it, and the information is still there. It's just lost resolution. And to me, we're part of that grand hologram.
Okay. I can give you a theory of the construct of the universe and who and what we are.
Yeah.
60 seconds or less.
So, I'll say this. Some would say that we are eternal beings that are temporarily occupying a physical vehicle for the soul learning of that vehicle. They're all part of a unified fabric of unity consciousness that are temporarily not seeing that because we identify ourselves as being the vehicle, the body, and not what we are is really ultimately the spirit. And that the ultimate result for everyone. And I love this particular quote from my esoteric writings, which was, we are all doomed to perfection, which means no matter what we've done, who we've been, who we think we are, that the ultimate end of this whole process of all that we experience is to return to a state of unity consciousness or being one with that infinite, ultimate, eternal love being of infinite knowledge, bliss, power, wisdom, and a never-ending experience of supreme bliss. So that is part of it. And a theory is this. If you understand the concept of the zero-point energy, which is one of the most extraordinary concepts even held by modern physics, is that the vacuum is not a nothingness, it is an unbelievably frothing, seething eruption of virtual particles in every direction all the time. It's
coming and going.
Flux. And if we took a cubic inch of the deep vacuum of space from every single point, and points are really teeny tiny, as you know, you can fit an almost infinite number of them in a small space. At every single point, you have this issuing of virtual particle fluxes in all different directions, all different energies, all different amounts of time. If they don't cross a particular threshold that's conditioned by the function of the delta T, the amount of time, times the delta E, the energy. If it doesn't cross a threshold conditioned by Planck's constant, we don't perceive it in our reality. And the math is, if you took all of that virtual particle energy and convert it to physical energy, and convert it into mass by Einstein's equation, that would create more mass than the whole known universe. So this virtual particle flux, this sea of energy in which we reside, the whole physical reality we see is the tiniest infinitesimal fraction of the energy that's there. And what condenses all that virtual particle flux into matter, my theory, is that the combined consciousness of all sentient beings in the universe creates one gigantic holographic grid, which condenses this very small amount of physicality from that massive energy density into what we call physical reality. That's a theory.
Pretty good for 1 minute. Lucas, I think you bit off a lot here.
Yeah.
Probably a little bit much for this podcast. Todd,
Not this last.
I am really deeply grateful for you giving me your time and allowing us to do this podcast. I would like to give you the opportunity to let the viewer know how to get in touch with you, how to contact you if there's doctors interested in training, because you indicated that you are training doctors how to do this. And the amazing thing to me was this doesn't involve bone marrows, this doesn't involve fat scraping, this is a blood draw. So it could be done in most any office. Do you want to give the viewer how to get a hold of you and either professional or lay person for treatment?
Sure. So the general office number for those interested in possibly experiencing the protocols that we do, including the biohack stacks, biooptimization, is 760-405-1908. For those practitioners that may be interested in learning more or possibly bringing this art and the sawing laser technology into their practice, the contact number is 760-405-4993. For general information and to see some but not all the full text articles till we upload them, there's a general site, drtoddo.com, drtoddo.com. And because I'm going to be in a movie called Biohack Yourself that many people will see, I decided to bite the bullet and actually have social media accounts. So I'm on Instagram at #DrToddO.
Very good.
I've just resisted it for a long time, but I guess it's time to get more information out there.
Really good. Lucas, do you have any final questions for Dr. Todd? Todd, I probably am going to want to have you on again sometime, but this was a blessing of information that's going to clear up a lot of mysteries of stem cell therapy, exosomes, etc. And I would imagine our audience will be as stunned as Terry and I were when we first heard you. So I thank you for it.
Great pleasure and honor being with you, and I love the work that you've done so well and courageously.
Thank you. Well,
Yeah,
you've got a
thanks.
lot of courage. You got a lot of courage to do this too, in the face of massive resistance on government, professional front, every front.
Yeah, there's that.
We're in the same boat, just different compartments of the boat.
Yeah, true. Yeah.
Lucas, any final comments?
When does this movie come out?
It's called Biohack Yourself, and the projection is it will come out in December on Amazon. And there's dozens of pioneers in the longevity and biooptimization. Well, I like that better than biohacking, actually. And the filmmakers have produced a film that has had 99 million views. I think this may even exceed that. Their goal is for this to be the most important health documentary in history. It has lots of people, Daniel Amen, Dave Asprey, Dr. Bruce Lipton, a long list of people that many from you know that are stepping out to give good information. So that is going to hopefully be seen by a lot of people and give them the information that they can become more of their own biologists than just listening to the narrative.
Yeah, maybe we'll have to get back together after or shortly before it releases and talk about it or build some hype around it and
Yeah.
give us some teasers on it.
Oh, sure. We're initially going to have just a documentary. The actual amusing one, the producer is Anthony Lally. He did a documentary on himself where he described how he lost 140 pounds in 9 months and became a champion bodybuilder, called Fat Lolly to Six Pack Lolly. 99 million views. So they know how to get a big viewership. They have a big following already. And they are really aligned with supporting us in terms of sharing all the media that they filmed. And initially it was going to be just one documentary, but now I think it's going to be in 6 parts. There's so much information.
Yeah, very exciting.
Yeah, cool stuff. Raising the awareness bar. Let's hope people listen. And
yeah, it starts here.
Yeah, exactly.
Okay, time to wrap up. Once again, I thank you. I know Lucas thanks you, and I hope that we'll be in communication.
Absolutely. Yeah, thank you so much.
Thank
I want to brainstorm more with
pleasure.
what you do and what we can do and get the word out and get better results.
Thank you.
As we conclude today's episode, consider the long-term health investment with Simply O3's ozone therapy systems. Designed for easy and safe home use, the Simply O3 3.0 Complete Kit comes with a lifetime warranty and a 6-month money-back guarantee. That's right, try it risk-free for 6 months and experience firsthand the reduction in brain fog, inflammation, and more. If you're not satisfied, receive a full refund. Ready to take control of your health? Visit simplyO3.com. I'd also like to give another shout out to our sponsors over at shopc60.com. If you haven't heard of C60, it's that antioxidant known to enhance mitochondrial function and combat inflammation. Many seeing C60 with their keto and intermittent fasting regimens, seeing a boost in energy and clarity. If you're past 40 and looking to refresh your vitality, explore shopc60.com and don't forget the 15% off code in the description for your initial purchase. Thank you for listening to this conversation with Dr. Todd Ovokaitys. If you found this conversation inspiring, please share it with your friends, family, and colleagues. Until next time, stay safe and be well.